Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 190 seminar, and I call it a University of Wisconsin Medicine Day. February 19 today is the 50th day of the year, and February has 10 more days to go. We have 10 more seminars to host to reach the 200th seminar, which will happen on May 21, 10 days after my birthday. Sometimes I am fascinated by numbers and coincidence, and I started to believe in fate after my miraculous recovery or survival from my many years of dying. Although we do make choices to partially control our and others' fate, I do hope we will make better choices regarding career, partner, proposal topic, awardee, our politicians, finance, place to live, and life. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Jean-Michel. He is a professor at University of Wisconsin Madison. He is originally from France and made a career decision or choice to work in the United States. I believe that decision was a great one, given his amazing contribution to the scientific community and education. Notably, he invited me last year as a speaker for distinguished lectures in microbiology series at his university. Of course, his university has the best microbiology program, and I really thank him for the wonderful time I had last year. That was Thursday, exactly 22 weeks ago, and I would be shocked if that was exactly 20 weeks ago. Professor Anne, thanks so much again for the invitation and your time today, and virtual podium is all yours now. And thank you again.
Thank you. It's my pleasure to be here. So I'm going to share my screen. Okay. Can you see it?
Yes.
Great. So I'm going to talk briefly about our work on the bacterial side to try to improve associations between nitrogen-fixing bacteria and crops, and in particular cereal crops for more sustainable agriculture. So I wanted first to talk briefly about what I call the nitrogen problem in agriculture. And as you probably know, most of our crops use nitrogen from the soil, mostly in the form of nitrate and ammonium. And what we call nitrogen fixation is the process of converting dinitrogen from the air into ammonium. Industrial fixation is the process that we use to produce synthetic nitrogen fertilizers. And these days, about half of the world population is sustained through the use of these fertilizers. These fertilizers have been used extensively, especially since the Green Revolution. So on one hand, we are extremely dependent these days on nitrogen fertilizers. At the same time, you may have heard that they have massive environmental impact and also economic impacts. The environmental impacts is, for instance, the nitrogen leaching, fertilizers going back into groundwater, leading to eutrophication of streams, rivers, coastal zones, but also the conversion of these fertilizers into greenhouse gases. And so a second process I'm going to talk about today, or what I'm going to talk about today, is biological nitrogen fixation performed by bacteria and archaea. And you may have heard that some of our crops, so the legumes, are associate efficiently with some of these bacteria and called rhizobia. And that's why we have legumes in all our cropping systems around the world. Unfortunately, when it comes to cereal crops, cereals like rice, maize, sorghum associate very poorly usually with these bacteria. And so it has been a goal of my community for decades, actually for more than 100 years, to improve association between cereal crops and nitrogen-fixing bacteria. So approaches in the community are sometimes on the plant side, trying to find a better plant, a better host. But today I'm going to talk about approaches on the bacterial side. And so the idea is to improve nitrogen-fixing bacteria associated with cereal crops. For me, in my lab, it's mostly these days maize and sorghum, but clearly we can apply the same technology and approaches for rice. So I'm going to talk about three approaches. One is what I call inducible ammonium excretors. We already, over the last years, over the last 10 years, worked on genetically modifying nitrogen-fixing bacteria, again, associated with cereals, like Klebsiella varicola, Enterobacter, Azospirillum. We already engineered these bacteria to fix nitrogen constitutively and release nitrogen outside of the cell. Very briefly, in terms of molecular targets, we affect the regulation of the nitrogenase. We basically make these bacteria blind to nitrogen in the environment. We decrease ammonium assimilation by playing in particular with the activity of the glutamine synthetase, and we limit the ability of these bacteria to capture nitrogen from the environment by knocking out ammonium importers. And so using all these strategies, we have developed what we call constitutive ammonium excretors. But the more we engineer them to be good ammonium excretors, the more we have a fitness defect. And so I'm going to talk briefly about the idea of having good ammonium excretors, but only in an inducible manner. Second approach that we follow is try to have better colonizers, so have improved the ability of these plants to colonize better our target crops, and ideally to target more the crops than, for instance, the weeds in the field. And the third approach that we have is finding bacterial helpers or helpers in general, but today I'm going to focus on bacterial ones, that by understanding microbe interactions in the rhizosphere. Because when we take a foreign microbe that we grow and culture and we use as an inoculant, very often the major pitfall is the inability of these inoculants to compete with the endogenous microbiome. So conclusion, what we try to do is that to try to engineer nitrogen fixation and produce these ammonium excretors, ammonium excretion, of course, because we want to share the nitrogen with the plant and improve the colonization of the plant. So first, the idea of inducible ammonium excretors. So a very classical approach that we follow to make the bacteria blind to nitrogen and have constitutive nitrogenate activity is to delete a gene called NIFL and replace that NIFL, a negative regulator of NIF genes, with a strong promoter that then drives the expression of NIF-A, which is a positive regulator of NIF genes. This way, NIF genes are expressed constitutively in a manner independent of nitrogen. What we do here, the idea is to do that in an inducible manner. And instead of putting just a constitutive strong promoter to knock out NIFL, we replace that negative regulator with a biosensor. And so for that work, what we use is one of our favorite microbes, Klebsiella varicola, that we isolated in my lab from sorghum. It's a non-pathogenic Klebsiella. It's really easy to manipulate genetically. It's an excellent fixer. And we already had a good number of biosensors in Klebsiella that respond to root exudate, and that work is a collaboration with Brian Fleger here at UW-Madison. And so, for instance, to show you that it works, I'm just take one of these biosensors, but now we have actually a good number of them. You are probably familiar with the arabinose biosensor. Well, it turns out, luckily for us, root exudates contain a good amount of arabinose. And so we can use that classical arabinose biosensor as an example. And so if we replace NIFL with an arabinose biosensor and we make another mutation, we delete the AMTB ammonium importers, then what we can create are bacteria that show increased nitrogen fixation with increasing levels of arabinose under low nitrogen conditions, which I would say is relatively easy. But what is even more exciting is that even when nitrogen is present in the environment under high nitrogen, Here, that column here is the wild type bacterium. High nitrogen levels repress nitrogen fixation in the wild type, but we can still have good nitrogen fixation and inducible by arabinos, even in presence of nitrogen in the environment. So that's one, but of course we want the bacteria to share that nitrogen outside of the cells. So here I'm showing you ammonium excretion, showing you that basically it works. Ammonium excretion of the wild type bacteria. And here in white, these are the bar graphs for ammonium excretion of our arabinose inducible bacterium. And here we show that with increasing levels of arabinose, we can release more and more ammonium outside of the cell. So we were able to achieve inducible fixation and ammonium excretion in Klebsiella, and with arabinose here is just an example. We had a lot of success also with fuminic acid and other biosensors. So that's one approach. Another approach I was mentioning is to engineer colonization. And here it's just to show you one example of what we have published recently, again, collaboration with Brian Fleger and Maya Venkataraman was a grad student in his lab. And so in collaboration with Maya, what we did is that we identified a protein. It's actually an outer membrane lipoprotein that was annotated of unknown function that we identified by comparing a pseudomonas that attaches well to maize roots to a pseudomonas that does not attaches well to maize roots. And so we found that lipoprotein of unknown function. And when we introduced that protein that we call MAM-A into different bacteria, Pseudomonas putida, that was the poor fixer in the first place, but also our favorite Klebsiella varicola or Studzerai, which is close to it's Pseudomonas, we can increase the attachment of these different bacteria to maize roots. So it's one approach for us to try to, and one example on how we try to improve colonization. A third approach I wanted to talk about briefly is microbe interactions. So I'm not going to have time to talk about bacteria-fungi interactions and my favorite arbuscular mycorrhizal fungi. I'm going to talk about just bacteria. And so what we do for that to try to understand how our nitrogen-fixing bacteria interact with bacteria in the rhizosphere, we use a synthetic community. So for instance, Claire Palmer here in my lab focuses on maize. And so she designed a 12-member syncom containing 7 bacteria that don't fix nitrogen from the maize rhizosphere and 5 of our nitrogen-fixing bacteria. And so what we did is that we tested the whole community, what we call, of course, the single individual members, but we also tested what we call the leave one out communities, where we remove one member. And we also tested all the pairwise interactions within that community. And the phenotypes that we recorded with all these different communities was abundance of the different members, which is very classical for syncom work. But we were interested, of course, in our outputs for us, which is nitrogen fixation that we evaluated using acetylene reduction assay and ammonium excretion. And with that all information, we basically built a model of how these 12 bacteria interact together. We actually went back using that model to experimental work to try to test intermediate level communities to refine the model. And so now we basically have a very nice model on how these 12 bacteria interact with each other. As a summary, what we observe, not so surprisingly, is a lot of competition between our diazotrophs and the native community, but also sometimes some synergy. And right now we are focusing on the synergy, meaning sometimes some bacteria from the community help our nitrogen-fixing bacteria for growth or nitrogen fixation. And of course, from a practical standpoint, it's very interesting because we can think about taking these bacteria and adding them to the diazotrophs for application in the field. But also, of course, we're interested in mechanisms. How do these helper bacteria help the diazotrophs? So that's the third direction I wanted to mention. First, I wanted to finish. I wanted to show you basically where we are at now. So clearly, we don't have a legume yet in cereals. Legumes get 80-90% of their nitrogen from rhizobia. In cereals, it's not that efficient. But last year, in collaboration with a company called Pivot Bio that commercializes some of these bacteria, we published that we can replace about, I would say, 40 pounds per acre out of about 200 pounds per acre of fertilizer in maize. So right now, yeah, you see we can replace about 40 pounds per acre out of 200. It's not so it's just a small fraction, but it's progress. What is interesting is that we don't observe any yield penalty. The main reason is that we think that the plant anyway produces these root exudates to feed the bacteria. So there is no reason. I mean, we don't observe any yield penalty at all. But the issue of that approach is that the environment matters hugely on the result. And that's, I would say, the main challenge I want to highlight, and where I think synthetic biology is really highly needed to make progress. Because what we are trying to play with is sometimes what people call the disease triangle, is that we have the environment, the plant, and the diazotroph that we're trying to engineer, but we also have the native interactions with the native microbiome. And so environment, plant, diazotroph or engineered bacteria and microbiome all interact, or it's what I call would be more the disease pyramid, if you want. And trying to understand better let's say diazotroph for me, microbiome interactions, and the impact of the environment and the impact of the plant genotype is really the challenge. All right, I'd like to finish and acknowledge my main collaborators for synthetic biology and synthetic communities, Brian Fleger, Carsten Zengler, and Ophelia Venturelli, and the main people in my lab who worked on that project, who are working on the project, Valentina Infante and Claire Palmer. With the time I have left, I just wanted to say that I'm looking for 2 postdocs to work on SynCom and synthetic biology. So if people are interested in the room, I have actually 2 openings that I'm looking forward to fill. Thank you very much for your attention. I would be pleased to take your questions.
Thank you so much. I mean, you know what? I believe in the future, the most important topic of research, one of the most important topic is, of course, crop, yield increase by nitrogen fixation. And of course, the second one is the microbiome research. And then you are doing both of them. I used to do nitrogen fixation using cyanobacteria, but I understand how challenging that project is. So I now kind of a little bit reduce my effort toward that one, but this is a fantastic work. And then another coincidence I want to say is I saw Maya yesterday during the EBRC meeting we had. So
Okay.
it's kind of
Small
another
world.
coincidence. Yeah, small world. I mean, I believe I know her. I mean, a few times I met somewhere, but I just saw yesterday around this time. I mean, so it's very interesting. So thank you so much.
Welcome.
I hope you also succeed in that, especially nitrogen fixation work with the people of Bio. I believe this company is one of my favorite company. My friend actually started a company, and that they are doing amazing job, implementing the nitrogen fixing bacteria in the U.S. soil and also Africa. That's a very important work, I believe.
Exactly. Yeah. I can also say that currently we focus on nitrogen fixation because it's a phenotype we can easily quantify, or we can quantify well, in particular in culture and also on the plant. But the kind of approaches that we follow, I think, can totally be applied to other benefits that microbes can provide to crops. So
Absolutely. Absolutely. So thank you so much. I mean, I hope, this one, benefit, a lot of farmers, including developing countries.
yeah.
Okay. So thank you so much again. Okay. So now the main speaker of today with a longer introduction, Professor Rosa Santomartino is an assistant professor in the Department of Biological and Environmental Engineering at Cornell University. So she just joined last year, so congratulations. She is a space microbiologist and biotechnologist with a deep interest in understanding microbial behavior under space conditions. This is one of my favorite topics because my last time is Moon. Her research focuses on leveraging this knowledge to advance sustainable space exploration and to address pressing environmental challenges here on Earth. That is actually, I believe, because the problem in the space applicable to the problem in the planet, this planet. She earned her PhD in molecular biology from Spinza University of Rome, meaning she made a similar choice to that of Professor Anne and me, working in the United States now from another country. During her doctoral studies, she completed internship at Claude Bernard University Lyon in France. That's another coincidence that is the country, Professor Ne actually used to be, and also at the Universidad Nacional Autónoma de Mexico, earning the title of doctoral Europaeus. In 2018, he joined the University of Edinburgh, UK, as a postdoctoral research associate in microbial astrobiology. There, she led two groundbreaking space microbiology experiments, BioRock and BioAsteroid, conducted on board the International Space Station. So she was all over the place, probably except the ISS, International Space Station, because I heard she just working with bacteria on board in the space. This brought her to collaborate with the NASA and European Space Agency. Her expertise was also instrumental in the development of Italian Space Agency roadmap for space life sciences. In 2022, she became a co-director of the UK Centre for Astrobiology at the University of Edinburgh and received a prestigious Leverhulme Early Career Fellowship. At Cornell, she is a member of the Carl Sagan Institute. Coincidentally, I will visit her country, Italy, and Dr. Anne's country, France, this year, very soon. But I have never visited International Space Station, although I have performed research on space bio and plan to send bacteria to ISS as she did. So I envy and respect her, and I'm so excited to listen to her talk today. Rosa, thank you so much for your time today, and please take it away.
Thank you so much. That's very nice introduction and make me sound so cool. So thank you. Can you see my screen?
Yes,
Okay.
please go ahead.
So first of all, really thank you for this opportunity of being here. I really appreciate the potentiality of the opportunity really of sharing my research through the SynBYSS seminar. And today I would like to tell you about my research in this innovative field, the field of space biotechnology, how this could not only enhance the sustainability of space exploration, which is a main goal of my research, but also pose benefit for Earth. So this is something that I hope I can focus, or I will convince you of how these two things are linked, hopefully in this talk. So the first thing to take into account, let me move this because otherwise I cannot see. Right. The first thing I would like to focus is the future of human space exploration, right? So in the last, in the 60s and the 70s, we saw that human exploration reached the moon. But then after that, in the last decades, we saw that it actually all focused around Earth orbit, right? Think about the International Space Station, the Chinese Space Station, in the past, the shuttle missions or the Mir station. And even in the next future, we are seeing how even with the close retirement of the International Space Station, some commercial space stations surrounding Earth are still, going to be present as efforts. So human space exploration surrounding Earth orbit is still going to be there. However, the future for human space exploration is, the goal, I should say, for the future is to move toward the moon and Mars and establish a permanent presence for humans there. Now, if you look at this image, I really like this image. I know that is not, the pixels are not great, but that's the best I can use. I like this because this image came from the International Space Exploration Coordination Group, and in the Global Exploration Roadmap from 2018. So if you look at that now, I used this picture for many, many years, and just when I was preparing for this presentation, I realized that because this is starting getting quite old as an image, you see how, according to this one, maybe it was quite positive because we are now in this timeline and we still haven't gone back to the moon. So maybe we are a little delayed on this, but I'm going to explain to you why that's the case. And the reasons for that is mostly the fact that there are many challenges that we have to address when we think about human space exploration beyond Earth orbit. Once for all, humans are going to require a lot of resources to make space exploration feasible. Not just stuff for basic life support systems such as food, water, and oxygen, but even in the long term you have to think about energy, shielding materials, even pharmaceutical compounds that you're going to need in the long term, and so on. Now, how does it work so far? Well, at the moment, because as I was saying, the majority of the space exploration is focusing around Earth orbit, for instance, think about the International Space Station, right? It's 450 kilometers above the sea level, relatively close to the surface, to Earth's surface. So any commodity that astronauts are going to need, like food, oxygen, hardware, anything, takes roughly 2 days of transit time to get from Earth to the International Space Station. And equally, if you need to bring something back to Earth, it's still 2 days of transit time. So maybe not the fastest, but still something that is feasible. And in this case, I always like to mention my postdoc supervisor, Professor Coquel from the University of Edinburgh, because every time we were talking about that, we were saying, well, that's roughly the distance between Edinburgh and London in the UK. So this is still a distance that is kind of feasible. However, if you want to reach the moon or Mars, then you have to consider an exponentially longer distance, which also converts to an exponentially longer transit time. Now, Professor Everest and colleagues, Professor Everest gave another talk to the SynBYSS, I think roughly 6 months ago, on a similar topic. They try to estimate a little bit the numbers. So when we talk about the resources that are going to be needed, how much are we talking in terms of mass, in terms of cost? And it's very difficult, but if you just focus, for instance, on the food, let's just focus about the food that is going to require to human crew that is going to reach the moon or Mars in the either long-term or short-term missions. Now, the numbers becomes, in terms of total cost, and we are just talking about the food needed, it can reach easily billions of dollars, but even if you just consider the lowest, it's still going to be in the level of millions of dollars. So you can imagine how bringing everything with you, every resources that you need with you, is not a feasible approach because it's not economically viable, but it's also not logistically viable because even if you ignore the cost and you just think about the kilograms, and again, just for food, it's prohibitive as numbers. So what's the problem? Actually, what's the solution? Well, if you want to achieve deep space, human exploration, we are going to find ways to achieve resources locally, to obtain resources locally. And this is a concept usually referred to as in situ resource utilization, which is a NASA definition. Now, the good news from me as a microbiologist, as a background in microbiology, is that microorganisms can really support us on this perspective. In fact, some microorganisms already have a lot of roles in producing different consumables, items, and they have a role in many industries on Earth, including in the food industry, in the pharmaceutical industries, for biofuel production, for the recycling of waste, and so on and so on. So because of their very important role for us on Earth, we also believe that they are going to be key beyond Earth, for instance, on an extraterrestrial settlement. And some of the key roles that microorganisms could play for us beyond Earth include not just the generation of food and oxygen, but even the recycling of waste, the obtainment of materials, and maybe in some bioindustries, for instance, for the production of pharmaceutical compounds on a settlement. Now, in my past research, particularly at the University of Edinburgh, but I'm still working on that now at Cornell University, one potential role, we were focusing on the utilization of microorganisms to obtain materials, which feeds directly in the concept of in-situ resource utilization that I just mentioned before. In fact, there is a microbial biotechnology called biomining, which utilizes microorganisms to extract metals of economic interest from rock ores, mine waste, and so on. And this is widely used on Earth to extract copper, gold, and other metals, including, for instance, rare elements. And it tends to be complementary to traditional mining techniques, but it also has been improving the rate of metal recovery from mine waste or mine heaps, for instance. And it tends to be considered generally more economic and environmental friendly. So there are many advantages of using this technology when possible. Microorganisms that are used in this type of technology include the chemolithotrophic microbes, but also organotrophic, including heterotrophic microorganisms. Fungi, for instance, are widely studied for that, and consortia. But there is also bioengineering approaches that now are under investigation to understand how we can improve these mechanisms. Now, from the perspective of the potentiality, I should say, to perform biomining in space in the last 10 to 15 decades, maybe something more, has raised a lot of interest, because if we can use microorganisms to basically obtain resources in space, we have a kind of a win-win situation because of the benefit of this technology. However, if we consider converting any type of technology, really, but particularly biotechnology, that works perfectly fine on Earth into a space environment, we have to take into account that conditions present in space are very different from those that we have on Earth. Organisms are adapted to the terrestrial condition, not to the space conditions. And I'm talking about, for instance, the different gravity conditions, but we also the higher doses of radiations, the lower pressure, and even the atmospheric composition that is different on planetary bodies or sometimes even absent, and also the extreme of temperature. So there are a bunch of conditions that we have to consider before understanding if we can transfer biotechnology from Earth to space. And this is something that I focus, as I was saying, my research, my postdoctoral research, and beyond. In fact, if you take into account even just the Moon, Mars, and asteroids, which are goals for future human space exploration, even just the gravity condition of these planetary bodies is lower compared to Earth. For lunar gravity, it's 0.16 G, Martian gravity 0.4 G, and asteroid we have something close to microgravity, which is usually referred to as weightlessness. And so we know that direct or indirect contact of microorganisms to the rock surface, to the mineral surface, is very important for biomining. So one question that we could ask immediately is like, okay, but how does gravity influence in which these microorganisms attach to the rock surface? And does this have any effect on the biomining efficiency? And this is something I had the chance to study again. And during my postdoc, I had the chance to lead together with the PI, Professor Coquelle from the University of Edinburgh, these two experiments on board the International Space Station, BioRock, that was launched in 2019, and BioAsteroid in 2020-2021. To clarify, I was not on the International Space Station. That would have been very cool. But as you can see from these images, we had excellent astronauts that actually take care of our samples when they were on board the International Space Station. So here you can see the ESA astronaut Luca Parmitano, and here you can see the NASA astronaut Michael Scott Hopkins working on our samples for us on board the International Space Station. And these two experiments had some similarities because the guiding questions were very similar. On one side, we wanted to ask, okay, how does gravity influence the way in which the microorganisms interact with the mineral in space? But of course, the reason why we wanted to ask that was also a biotechnological one. We wanted to understand how does this influence affect their biomining capacity. But there were also some differences between these two experiments. For instance, for BioRock, for both BioRock and BioAsteroid, we use the same bacterium Sphingomonas dissecabilis. But for BioRock, we use other two species. And we used a terrestrial rock, which was an Icelandic basalt rock that is an analog for moon and Martian rocks. While for bioasteroid, we decided to go for a bacterium and a fungus and a synthetic consortium of the two, and we used the real extraterrestrial rock. So as you can see, the goal at the end of the day is the same, but we were trying to differentiate a little bit and expand a little bit the information that we could get from these very rare occasions of experiments. So the first thing that we wanted to understand, of course, was, okay, can we have any metal extraction from the rock that we selected from when the bacteria are or the fungi are in space? And the results were quite promising. In fact, we were able to demonstrate vanadium, rare elements, and also platinum group element, particularly palladium extraction, on board the International Space Station, which was very good because it granted these two experiments the first proof of concept demonstration of biomining on a space station. However, something that I want to highlight is that the extraction level was relatively low. I think we had something that in the best case scenario, that was the palladium extraction mediated by the fungus Penicillium simplicissimum, the extraction was 10 to 15% of the palladium actually present on the rock. So not something that is still yet, commercially available, but still we were able to demonstrate that in principle the concept is there. And so this gives us some, insight on what we can do after that. But something that I want to highlight is that even if the extraction itself wasn't great, at the same time, when we compared the extraction mediated by the fungus compared to the non-biological control, we still had an improvement of roughly 600%. So something that tells us, okay, maybe it's not economically viable yet, but still the fungus give us an improvement, an advantage compared to not using that. However, not much is known about the mechanisms underlying that. There is really not much about that, particularly for the fungus. So what we had the chance to do for the second experiment, the bioasteroid experiment, was also having a metabolomic analysis that gave us a little bit of hints, a generalized hints, right? Not very specialized for different molecules, but a generalized hint on is microgravity somehow affecting, the secondary metabolite that this microorganism can produce? And the results, again, were quite intriguing because we saw that particularly the fungus, which you can see here and here, the fungus had an increased number of molecules that were expressed and produced particularly when they were exposed to the space condition compared to the terrestrial condition. Some of these features were of potential roles in bioleaching. Some other have a potential pharmaceutical interest. There is something, that's a promising result, but this is something that we need to investigate further. It just tells us that microgravity does influence the metabolic profile, and this was important results also because there is not much in the literature. There is very, very limited data about metabolomic analysis in space conditions, and particularly, I think this is the first one about bioleaching, metabolomic analysis for bioleaching microorganisms. But then, after having these good results, having these exciting opportunity to work in this field, I went back to the potential roles of microorganisms in space. And what I was really interested in understanding is which other of these potential tasks that microorganisms could play for us in space do I want to focus on? And I realized that I was really intrigued by the potentiality of using them to recycle waste in space. And why? Well, basically because I mentioned a lot how resources are going to be quite limited in a space environment, and how important it will be to obtain resources locally. But we are going to bring some materials with us. So if we can actually recycle them as much as possible, this helps us to, on one side, minimize the resupply of resources from Earth, while at the same time also helping reducing the production of space waste. So in turn, this will help not only achieving a self-sustainable human presence in space, but at the same time also addressing ethical considerations that are associated with the production of space on an extraterrestrial settlement. And so the next step for me was, okay, but how is waste disposal currently happening in space? How does it work? And the results were a little, I'm going to show you this video here, showing that at the moment, waste disposal in space is quite problematic, as you can see from these images here of astronauts, getting clogged really by all the big amount of solid waste that is produced on the International Space Station. Here you can see that usually solid waste is just compacted as much as possible, and then is just ejected toward the Earth's surface, which is of course a feasible solution when we think about orbiting Earth, but you can imagine how this becomes problematic if you are moving to the Moon or Mars, because we cannot do this anymore. Now, this is not, a novel issue. It's not something that I happen to discover. Space agencies and space companies are very, very well aware of this issue, and they are trying to solve it as much as possible. And a lot of effort are being put in the last decades or so toward how to recycle biological waste. And by biological waste, I mean food waste, human waste, wastewater, and CO2, for instance. In this case, I'll always like to mention the European Space Agency MELISSA project, because I think it, highlights very, very well how biological system can really support the recycling of biological waste for space exploration. And I like this project also because among the 5 compartments that you have in the plant, which is 1 compartment, second, third, fourth, and fifth, actually, sorry, first, second, third, fourth, and fifth, 4 out of 5 compartments include the use of microorganisms. So again, it highlights how they will become important. However, there is something that is missing in this equation, if you want, which is what about synthetic waste? When we are going to go in space, we are going to have also metallic structures, electronic devices, clothes, consumable plastic, a large number of synthetic waste, which is currently not considered as much. And this is surprising because it has been estimated that future payload for lunar or Martian mission, either in the long term or in the short term, are going to be constituted mostly more than 70% of plastic, metals, electronic, so all these type of synthetic consumables. This means that in future, synthetic waste is going to constitute the largest portion of future mission waste. And this sounds very ominous, like, okay, what do we do with that? But I think we should see this as an opportunity rather than a burden, and I'm going to explain to you why in a second. In fact, if you take into account, for instance, plastics or electronic waste, these are all materials that are rich in carbon, oxygen, hydrogen, metals, and other elements. So if we understand how we can recycle these all these precious elements, which are going to become an important, resource potentially in a space environment, we could create feedstock for biomass, for the production of biomass, consumables, and biosynthesis, for instance, in a space environment, while at the same time helping, again, the reduction of space waste production. And this is something that I am focusing mostly my current research at Cornell University, my laboratory that just opened in May 2025. I'm very excited about that. We are focusing mostly on understanding how we can develop these biotechnologies for synthetic waste recycling in a space environment. For instance, from the perspective of plastic, this is a project that my PhD student Melina Andrade is focusing at the moment. She is at the moment using microorganisms of known plastic biodegradation capacity. Particularly, she's focusing on Idonella sakaiensis that was discovered 10 years ago now, because it's quite well established as the mechanism for the biodegradation are well established. And what she's trying to do is understanding, so we know that this microbe can degrade PET, so polyethylene terephthalate, but how does the space condition affect the capacity of the microbe to degrade the plastic? And this is the first step. And then after we are going to understand this, then we can understand how is the efficiency better, worse, or equal? And which are the enzymes involved? Now, again, the enzymes directly involved with the degradation of the plastic in this microbe are well established, and it's basically based on 2 enzymes, the so-called PETase and METase, which are 2 hydrosylates enzymes. However, when we think about adding the space conditions into that, other factor might become important. So which are the other enzymes involved in this process, and can we bioengineer them in order to increase the efficiency for space applications? And at the same time, can we use the biodegradation product of the plastic degradation and to bioengineer Idenella sagayensis so that we can upcycle this product into useful molecules? This is something that is being demonstrated on Earth, particularly on E. coli. So we are trying to understand, can we do that under space conditions? And another approach will be, okay, what if Idenella sagayensis, like bioengineering Idenella sagayensis, maybe is not necessarily the best approach, because maybe we discover that it doesn't really like the space conditions for some reason. So can we actually bioengineer microorganisms with demonstrated resistance and with the demonstrated capacity to thrive under space environment and space conditions? And can we bioengineer these microorganisms to actually degrade the plastic and do everything that we want them to do? We are focusing a lot on Bacillus subtilis because it's a model organism which makes our life easier, but at the same time is also a microorganism that is widely used in space microbiology. So it will tell us, it should at least give us the perspective of, okay, it should not be a problem for it to thrive under space conditions. So these are some of the approaches that we're using at the moment. It's just a few of them. And then we are also focusing on the recycling of electronic waste. This is something that my PhD student Emma McGuire is focusing at the moment. She is using these fungus that maybe you recognize. This is the same fungus that we used in the bioasteroid experiment, so the second experiment that we launched on board the International Space Station. And why did we focus on these microbes? Because I was about to say coincidentally, but it wasn't a coincidence. We knew about that. This microbe has been demonstrated not only to perform bioleaching, so metal dissolution from rock and electronic waste, but it also a degrader of some type of plastic, particularly polyethylene. So what Emma is focusing on is trying and understand if we can use these microorganisms to degrade electronic waste, because the difficulty of recycling electronic waste, even in terrestrial setting, is the fact that these materials are very complex materials. They are made of metal, sure, but also of polymers, as well as ceramides and other compounds. So it's quite difficult to make the best. When you degrade them, you have to focus on one. What do you want to do? Do you want to extract the metal? Do you want to extract the plastic and all of that? And it's quite difficult. So what she's trying to understand is, can we harness the power of this fungus here, which can do both things, to link them together and create a better, degradation, an efficient degradation of the electronic waste? And then we want to understand, of course, we are space microbiologists after all, so can it still do the same thing under simulated space conditions? Is the efficiency equal, better, worse? And once we understand that, can we bioengineer this microbe to make it, if needed, make it better, or to limit the difficulty that it might have doing that in space? And another approach, of course, will be similar to what I was saying before for plastic. In this case, would it be another option to, for instance, utilizing just a bioleaching microorganism, such as, for instance, this bacterium, Clugonobacter oxidans, which has been, it wasn't considered, I don't think it's still considered a model organism, so it was quite difficult to bioengineer, but my colleague in Professor Barstow's laboratory at Cornell University, they managed to pioneer the bioengineering of these microbes. So now the idea is, can this microbe that has non-bioleaching capacity be engineered to also perform the polymer biodegradation, so that at the end of the day, it can do both things together? And if not, can we create a synthetic consortium between a microbe that maybe can perform metal dissolution and another that can perform polymer degradation, and so that we make this effort efficient? And the last thing that I'm focusing on, this is not really bioengineering in a way, but there is the potentiality for bioengineering. But I still wanted to mention that because I am quite passionate about this aspect of my research, is attempting to discover novel species with biotech potential from the environment. So to this aim, we performed a bunch of different field works, particularly in Scotland between 2022 and 2023, as well as Iceland. This one, I was not able to join this one because May was exactly the month that I was moving from Edinburgh to Cornell University. But the student Ines Pereira Figueiredo, which is a PhD student at Edinburgh, University of Edinburgh, she managed to perform this field work. And what we are doing, we are collecting plastic debris from this polluted environment. And the idea is to try and see if the microbiome that is surrounding this plastic debris can has some biodegradation capacity. It's a long shot, of course. This is not a given, but all the microorganisms that we currently know having biodegradation capacity, including Idenella sakaiensis, have been discovered by this approach. So this is something that I hope can give you some good results. And then the idea will be then after we discover these novel species, can we use them for biotechnologies? So that's a little bit what we focus on. And to conclude, I hope that I convinced you that synthetic waste biorecycling is going to be key for as a tool towards circular economy in space. This is a concept that we try to demonstrate with this paper from 2023, where we show that a waste recycling compartment on future space missions could really be the core, at the core really of future effort for circular economy in space and life support system and in situ resource utilization, settlements on compartment on extraterrestrial environment. But something that I am really passionate about is understanding how developing these loop closure, this very efficient loop closure in circular economy and technologies can benefit Earth. Because at the end of the day, circular economy is something that we want to try and achieve on Earth as well. And again, so once we develop these very efficient technologies, how can this benefit Earth? In fact, so far I spoke about waste, I thought about plastic a lot, and on those terrestrial perspective, but we are all aware about how we have the so-called plastic crisis on Earth. At the moment, only 9% of plastic is recycled globally. And this is posing not only an environmental threat, but also an economic loss. Some people estimated that you can lose as high as 110 billion of dollars per year by not recycling plastics. And this is very motivating for me, but particularly when over these years of working in the space, the space biology, the space sector, really, very often I got asked by the public, but why should we worry about space exploration if we have so many issues to solve on Earth? And honestly, I think it's a valid question to address. I think it's the question that we should address. So when I started getting asked this question, I started thinking, okay, is there actual benefit that space exploration, space science pose to Earth? And I discovered that the answer, I'm very happy about that, the answer is yes. Indeed, a lot of technologies that we currently use in the everyday life, GPS is the most famous one, but we can also talk about CT scans, high-tech artificial limbs, water purifying systems, freeze-dried food, and even portable computers. I'm talking from you now from a portable computer. So all of these is possible because of space exploration, because these technologies have been originally discovered and developed for space exploration, and now we have them available in our everyday life. And moreover, I also discovered that the United Nations recognized space activities as essential tools to achieve the Sustainable Development Goals. So by doing the research that I do, I realized that because of the limited resources and the stringent conditions of space really can act as a model system for developing these high efficient loop closure and zero waste technologies on Earth. So the take-home messages for my talk today is that, first of all, for long-term missions to the Moon and Mars, we are going to need very efficient life support systems and mechanisms to acquire resources locally, so the so-called in situ resource utilization technique. However, on this perspective, bio-based processes are particularly promising, particularly for life support system and in situ resource utilization again. But developing these technologies is not just going to help circularity and loop closure in space, but also pose great benefit for Earth. And with that, I would like to thank all the people that are involved in this work, the work that I presented so far. Definitely the people in my group at the university, at Cornell University. I added the PhD student Ines Pereira de Figueiredo, she's actually at the University of Edinburgh under Professor Coquel's supervision, but we collaborate quite strictly. So I still consider her part of my lab, even if from afar in a way. And all the people, definitely all the group, the UK Centre for Astrobiology team in University of Edinburgh, with particular mention of Professor Coquel, and all the people involved in the BioRock and BioAsteroid experiment, which are really, really a wide group of people. I would like to thank you all for your attention, and I will be very happy to take your questions.
Thank you so much. It's an excellent talk and exciting. Also, Ines is not far from you because that is closer than Moon.
Yes.
Fantastic. So, Jean-Michel, do you have any comment or question?
Sure. I was wondering, you showed one slide on comparing the efficiency of your microbes on Earth versus space conditions. What's the order of magnitude? Are they really 10% efficient in space compared to Earth conditions, or
let me see if I can.
Maybe you actually showed that already, but I was wondering how.
Sorry, I have a lot of extra slides, which
I'm sure.
But let me go to the slide, which I hope I have on. Oh, okay, here. No, actually. Give me a sec. Yes, I did have a slide, particularly. Oh, here it is. So,
oh, I haven't seen the colors. Yeah.
No, I just showed this just for sake of time, but we did the extraction. So we did our calculation. So this one, we were just focusing on the difference between, basically the percentage of extraction between the biological organism and the non-biological sample,
Yes.
just for International Space Station. While here you see the same data, but these are comparing basically the percentage of extraction from the rocks, from the meteorite. And here you have in green the International Space Station samples, and in orange the terrestrial samples. So here you can really see, that in some cases you have.
Yeah, it actually seems to behave even better on the ISS.
Yeah, it's interesting because one thing that we notice is that it's really, really depending on the metal that you are taking into account, the species that you're taking into account, and the rock that you're starting with. And I'm saying that because it's something that is quite difficult to explain when you give a talk, for instance, or even in the paper I had, my first draft of the paper was huge discussion, and then I have to shrink it. But the reality is that I think is a very interesting data because in the space microbiology field, there is always this effort. It's been, decades of effort trying to understand what is the effect of microgravity on microorganisms, and now we are trying to understand what is the effect of microgravity on bioleaching. And the reality is that the answer is always it depends. And I think with this type of experiments, it really highlights this a lot because we have, all these samples were subjected to the same conditions, to the same materials, the same rock materials, etc., and you still see that it depends. So, for instance, I think a good example is the palladium one. You can see that when you consider the extraction rate compared to the non-biological control, you have an increase of almost 600%. Yes, the variability is high, but it's still almost 600% compared to the non-biological control. So it tells you, well, actually, when you are in space, using the fungus is better. But if you compare the terrestrial versus the space, you see that actually there is no change in the penicillium capacity of extracting palladium. What changes is the non-biological one, because there is a massive difference between the two. So what this is telling you is that maybe in future, if we have to decide, we want to extract palladium from asteroids in space, shall we use the bacterium or not, or shall we use maybe a non-biological system? It tells you, well, it's not that in this case, it's not that the fungus has a massive difference in space, is that
because the non-biological is so inefficient.
exactly. And why is that? We don't know. I think one of the reasons could be that the fungi, by forming the mycelium, and we also tested how the mycelium is, if there is any effect of the mycelium formation on top of the rocks under space condition compared to Earth, and we didn't see any macroscopically difference. So maybe that's just their biology.
Let me comment one thing. So we only use the simulated kind of chamber on the Earth to do our work that is published in Nature Communication. But the problem of using ISS is it's very expensive. Once you've done some experiment, if you want to repeat similar experiment, you need to wait another, I don't know, several months. So this is not easy experiment to do. And then I do understand now, because why not just doing multiple more experiment, but that's not easy thing to do. So this is an important data set. But this is published, you said, in submission here.
Oh, sorry, this is an old slide. Yeah, now it's published on very recently, end of January.
Oh, that paper.
Yes,
Okay.
I think the slide that I showed you before, I managed to change it, but because this was the additional slide, I forgot to
Okay, so this is
update it.
probably supplementary, right?
No, this is in the main figure of the paper, but in supplementary we had, let me see if I can show you. In supplementary, so we didn't just focus on platinum group element. We actually had analyzed 44 different metals, which I wish I can find. Oh, here it is. And among these, then we did a little bit of what's the word I'm searching for? The statistical analysis to try and understand, okay, which one are actually of interest. Maybe I also have, no, I don't have the image of the whole analysis, that all the 44, but when we try to understand, okay, which one of these is actually of interest, we discovered that some of them are, and this is what I was mentioning before when I was saying, the results really changes depending on which metals you are taking into account and other things. And you can see that in the majority of cases, so except for sodium and phosphorus, where you can see in green you have the International Space Station, right? And the efficiency was better in space compared to Earth. In the majority of the other cases, the extraction was better on Earth compared to space. So why is it the case that for sodium and phosphorus you have this better extraction in space, but not for the other metals? I guess it's a combination of factors. It's probably due to the mineral that contains these microorganisms. These elemental species is probably the thing that is going to address that the most. But these also tell us that it's very difficult to predict. Okay, I want to biomine XYZ metals, which microbe in space is this microbe going to be worse or better affected by microgravity? Well, I don't know, because as you can see, in some cases it's better, in some other cases it's worse.
But you know what? My point is, comparing between Earth and ISS is important. To me, probably for the practical application, the most important thing is, as long as this bacteria working on par or similarly or better or a bit less, it doesn't matter. As long as this work, I mean, it's gonna be useful.
Yeah,
Of course, I
definitely.
mean, for paper publication, you need to show the comparison between, the Earth's condition and the microgravity condition. But as long as that work, I mean, that should be fine. That's my point.
No, definitely. Well, I think what I mean is that, like for instance, we know that Penicillium simplicissium can extract a bunch of metals, for instance, copper, yeah, for instance, well, copper, it behaved quite well, but other metals from on Earth. And so you could just assume, okay, it's going to work equally fine or even better, I don't know, in space. And some other time, sometimes the result is no, actually, if you make it do what you want to do in space, it's going to go better or worse. So I guess it's just a matter of, I think you're right that as long as it works is it's fine, but it's difficult to predict if it will work with the better, equal, or worse efficiency.
Right. Do you have any other question to her?
Yes, I was wondering, so the approach that you're currently following is testing individual microbes, and at one point you were talking about combine, well, no, actually, you actually combined them already. So it's really the bottom-up approach. I was wondering, what about the reverse? A bit your idea of going to that highly polluted site in Scotland or in the UK, and what about taking a whole community? How efficient is the whole community of maybe even uncontrollable microbes compared to individual microbes? Are they better or just no?
That's a very good question, and indeed, that's something that we are doing right now. My lab manager, Chris Derito, here in Cornell. So from the Icelandic isolates, so the samples are isolated from Iceland. We have both the community and now he managed to isolate I don't know how many samples from species from these samples. So we question a lot, shall we start from the community and see what happens, or shall we start from the isolates? Eventually, now we are trying to use just the community to try and see what is it that is enriching. And now what we are doing is that because we also have the isolates from single species, basically we have a sample where we see good results. So now we are saying, okay, let's try and see what is enriched from the community. And at the same time, let's try and take the single isolates. And what I would like to understand is is it exactly what you say? Is it a single species that is doing the hard work, or is it actually the community? So then if you isolate them, you don't see anymore
Yeah,
the degradation.
you can
And so
take the
yeah,
problem
that's
both ways, bottom up and top down.
exactly, yes, yes. So that's part of that. I guess when you start from a single species, like what we are doing in this type of experiment, yeah, if you start from an environmental sample, yes, sure, you can start from the community. But because on the bio asteroid experiment, we were kind of doing the opposite. We were asking, oh, can this microbe and this other microbe do that? Then it becomes a synthetic community, which is
Mhm.
not necessarily the best approach. Yeah.
Great,
Um,
awesome.
Any
Another
other question
question
you
out
have?
of the box. Maybe go ahead. Go ahead. Go ahead.
No, you have any question more?
No, I was wondering, what about bio waste, like recycling human waste?
mm,
Are people working on that? I
yeah.
know a group back in France working on using lactobacilli to convert human urine into fertilizer for crops. Uh,
Yeah,
is that what do you think about that approach? Or
I think it's a very, very important approach. And I mentioned at some point the European Space Agency project, Melissa. What they are doing is focusing mostly on this type of approach. No, not just them. Like European Space Agency project is the Melissa project is actually using a different, bunch of people across Europe, really. So it's a project that it encompasses many people, many groups around Europe. And I know there are also a lot of people working on that, definitely in the US and I guess everywhere. I think it's very valuable because one of the most important things that is going to be needed in space is food, as I said, which at the moment translates particularly in plants. So if we manage to understand, hey, can we use microorganisms to for instance, extract the nitrogen from the yellow water from astronaut and try and convert it to grow plant and even the phosphorus and all of that. So that's a very, very important approach. The only reason why I'm not focusing that myself is because many people are already focusing on that. And I think there is a gap towards synthetic waste recycling. But it's very interesting, and I keep on being contacted by people that want to kind of link the two things because eventually what you could do is that I don't know, you use now I'm just making an idea like out loud, like if you can take a bacteria, even fungus actually, if you can utilize a fungus that produces biomass starting from plastic waste, and then you can maybe this is something that you can use for improving root, right? Like in the rhizobium, here you go, you have a beautiful win-win situation in which you take into account both waste materials together.
Yeah. If I can mention that I have two projects with NASA these days in collaboration with Simon Gilroy and John Baker. One of them is looking at the impact of microgravity and cosmic radiation on microbiomes associated with plants. And so
Beautiful.
that one is, and the second one is looking at nitrogen fixation on the moon or in the moon regolith. So
Yeah, that's
I have
very
some interest
nice.
in base applications of nitrogen fixation.
So
It's so,
yeah, you know what, as I kind of briefly mentioned during my presentation in the Wisconsin, we three of us working together, I mean, I believe. So what we demonstrate recently in the paper we published in Nature Communication, basically exactly what you just mentioned, we used fecal matter. We want to use human waste, human fecal matter, but at the time, regulation, very complicated. So we actually used the so-called dog shit. We use dog shit basically, and then we use that dog shit to be processed by the NASA people, and then we get all the nitrogen source from that. And then we use the regolith, basically mineral from the moon or Mars, but we also use the composition of that one that we know because the regolith is very too expensive. So what we did is we actually add those two waste, or the one waste and one the in situ resources, and then we actually use the plastic PET, and then we demonstrated bacteria perform almost same as the Earth's gravity condition. So that is actually we published already. So I'm happy to collaborate both of you regarding those space issue or problem. And then original problem we wanted to solve is we wanted to use the photosynthetic bacteria, actually even fixed carbon. And then one thing I want to add is the nitrogen fixing bacteria. In that way, we have the really, the in situ biomanufacturing without relying on anything else except just in situ resources. So that's one thing I really want to do.
Yeah, definitely. And I know a few group of people that focus on that. The first person that popped into my head is Professor Verso from Bremen University that works a lot on cyanobacteria and even biomining, right? Utilizing cyanobacteria to perform biomining and bioleaching in space to then address in situ resource utilization and even life support system. And I put this back in the sharing because when you were saying that, I see a connection between the three of us, I agree. First of all, I took a lot of notes of Professor Rane when you were talking because I thought, oh, I have to ask about this, I have to ask about this. But if you adjusting this image here, you see here there is food and medicine production, and you see how you have plants here, but you also have the microorganisms that are supposed to represent the rhizobiome here. But then you have the regolith here, and then you have the human habitat and laboratories. So there is waste coming from the astronaut that can go into the life support system. So this is just an idea of everything is going to be connected eventually.
Exactly, yeah.
Absolutely. So that's how we make that project working. Actually, NASA people actually work for us, basically taking the fecal matter and then process anaerobically and then generating nitrogen source for us. And then we just mix with the plastic and regolith at the time, the mineral from the Mars or the moon condition. And then we use the bacteria that, basically produce the compound lycopene out of that, the condition. So we are very excited. That's my favorite paper actually, because I wanted to go to the moon one day because that's the only place I never been to because I'm traveling a lot, but I need to go there. But unfortunately, last year, I just almost get the ticket to send my bacteria, but unfortunately, NASA heavily, you know, you know, just hit last year, so we couldn't send. So I have a question to you. So how you just access to the this ISS? You basically contact the NASA and then
No.
somehow get something out, or how you just send the bacteria now. I mean, now you are in not UK, but now in United States. How you do?
Right. So, well, I'm not familiar. I'm not too familiar with the U.S. system, so I can tell you how this happened when we were in Europe. And the first experiment, BioRock, from when the Professor Coquel initially, proposed the project to actually when it went to space, it took 10 to 11 years. Normally, it doesn't take so long. I think there was maybe 5 years of kind of a gap because nobody was actually working on that. So we can maybe shrink it to 5 to 6 years from when you actually propose that to when it actually gets to space. And I think it just went to a call, like an open call for European Space Agency for, they were asking for ideas to proposing project really to send experiment on the International Space Station, and his idea was selected, and so after that it escalated, the process faster in the sense that when then your project is selected, then you can start doing all of your preliminary analysis, and then you start getting grants to do all the things. So that was through European Space Agency. For the second experiment, it was actually faster because we went through a commercial route, basically. It was Kaiser. Oh my God, I should know the name. Let me go to the name because it's important. Bioreactor Express from Kaiser.
Mhm.
And I think it's in the logo here, although this is very small. But yeah, Bioreactor Express. So this is a commercial route. So basically, after I think we contacted them in 2020, and in 2021 our sample was already in space, because in this way, basically they already have everything. Well, I don't know the details too much, but in this case it was faster because the hardware that we developed for BioRock was already developed, so we could reuse it for bioassay, which shrink massively the amount of time needed to run the experiment, but also because by doing through the commercial route, basically Bioreactor Express already had everything, all the links that were needed, all the communication with ESA and NASA and UK Space Agency and whatever was already on board. So there is an aspect of what happens behind the scene, which is difficult to, at least for me, to fully grasp, just because it's very, very complex thing, right? Like it is not. I keep on saying to my students, because my students, of course, have a high interest in having experiments launched in space. And I keep on telling me, are you sure? Not because it's a fantastic experience. I loved it, but it's also way more complex than just having a flask in your bench and you can repeat the experiment as many times as you want. And if something goes wrong, you just run it again, and all of that. So I think now it's getting relatively quicker to get experiment in space, but I think you still have to go through directly or indirectly space agencies that will eventually allow your samples to go
Sure,
to
sure.
space.
That's all. I almost there, but last year, things kind of not worked, because
Mm-hmm.
we have everything already tested in microgravity condition with the NASA people here. So we almost got that, opportunity to send my bacteria to the ISS. But last
Mm-hmm.
year, we heavily hit, especially NASA heavily hit. So we couldn't do so. So now I'm looking for the private sector. So I went to
Mm-hmm.
Korea. They invite me to give us some invited talk, and then there are some company actually, probably willing to send my bacteria if I get the grant, of course.
Mm-hmm.
So I'm
Yeah.
looking for those kind of out, but it's not easy. I mean, it also takes time. So
Yeah.
the
Yes.
problem is, let's say one student working on whatever space project, or postdoc working on space project, and then if that takes 2 years, and then they probably already left. I mean, so that's the problem we have. So
Sometimes you also have the scrubs of the launch. So, for instance, for the first experiment, BioRock, the first launch was scrubbed just a few weeks, not massive amount of time. So there was the risk that we had to re-prepare the samples. Now, because our samples are microbes and they were resistant to desiccation and stuff, it wasn't a problem. But sometimes when you work with human cells or cell cultures,
right.
it's very important that you prepare the samples just immediately before the launch. And if the launch is scrubbed, then you have to repeat all the preparation.
Right.
So it's not the easiest, but I think in future it's going to get easier.
Yeah.
We are going to have more opportunities. I hope it gets easier.
Yeah, there are sometimes they cancel the launch because of the weather conditions. So
Yes.
that is also very, So this is I do understand, and then this is not an easy problem, but I want to solve this problem, and then I want to do something in the space, because my dream is actually really going into the moon. I mean, one day in my lifetime. So do you have any question? Because I now see the 11, almost 30 Central Time. Probably we need to close officially, and then we could chat more after that. Okay, got it. All right, so let's close. Thank you all for joining and staying today. We'll meet again on February 26th, Thursday, the same time, the same Zoom link. We'll have the Professor Virginia Cornish at Columbia University and Abhishek Gupta at UC San Diego and J. Craig Fenton Institute, the Mine Institute. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists. And thank you. And I stop recording 1 second.